All right, let's think about meiosis. This is a cell. As it's preparing to enter meiosis, it will go through S phase and make a copy of each of its chromosomes. In this cell, I am just illustrating two chromosomes, that two homologous pairs, one that has a black centromere and one that has a brown centromere. So let me finish with my S phase. There's my S phase, and I'll go through G2, and now I'm ready to start meiosis. In meiosis 1, the chromosomes are going to coil up so we can see them, and that's when they pair. So each homologous pair finds its buddy, and they pair up. At that point, they'll do the little crossover thing. They'll exchange some genetic material. So this is prophase 1, where homologous chromosomes are pairing and exchanging some material. Let's finish that. For metaphase 1, the homologous pairs will travel together to the metaphase plate, line up in the middle of the cell, and the spindle will form, nuclear membrane has dissolved in prophase. Centrioles line up at either side of the cell. The centromeres hook up to the spindle, and homologous chromosomes are pulled to opposite poles. Now, there's been some confusion over the term independent assortment. That simply means that each homologous pair lines up how it wants to, independent of how any other homologous pair lines up. So if I got this magenta chromosome from my mom and the blue chromosome from my dad, I could have the blue go to this pole, the magenta go to that pole. They are equally likely to line up so that the magenta goes to this pole and the blue goes to that pole. And that's true for each of my 23 pairs of chromosomes. So if I originally got the white and the magenta from my mom and the blue and the yellow from my dad, they might separate at this first division along those lines, or they might not. So that's independent assortment. They move independently of each other. All right, so when they arrive at the poles, we go through telophase, and let's see if I can get a couple telophases here. Nuclear membrane rebuilds, chromosomes decondense, recondense, and now I'm ready for prophase 2. After prophase 2, in each of my new cells, here we go, my homologs now, my chromosomes will line up at a new metaphase plate. Spindle will form each of my new cells. Here they go. And chromosomes will line up with their centromeres attached to the spindle. At this point, the centromeres will duplicate and the sister chromatids will separate and move to opposite poles. So here's one sister chromatid heading that way, the other heading this way. I'll duplicate this centromere. One's going this way, its sister chromatid goes the other way. Same thing happens over here. Duplicate my centromeres, head that one off toward that pole, and same thing over here. This is now anaphase 2, where we're separating sister chromatids, and when they finally arrive, we'll move into telophase 2 and end up with 4 nuclei. This will dissolve the spindle. That goes away. And now I have 4 nuclei. Each one is haploid, has 1 copy of each of my 2 chromosomes. If this is in a male, these will become sperm. Each one is a little different than the others, and they're ready to go off and potentially fertilize an egg to make a zygote.